Hi everyone. Today I'm going to be talking about supramolecular hacky sacks, how are they formed, and some concepts that we may need to understand the whole system. So let's look into what are they. These are nanoparticles that we've developed at the University of Puerto Rico, and so far we've been able to encapsulate small molecules, proteins, and plasmid DNA. Therefore, we have a very strong and flexible tool. Now that we have this tool, we started to implement in different applications. And what I'm showing you guys here is the encapsulation of dextran 6 red, which would be the red dots, not Teresa, the red dots around the nucleus of the neuroblastoma cells. So here we can see in this confocal image that they don't only encapsulate, they go inside the cell and they somewhat get to the area of the nucleus of these type of cells. So let's look into the formation of these hacky sacks. The hacky sacks themselves are the thermal response of supramolecular guanosine quadruplex. Let's first look at that process, the thermal response. It's called the lowest critical solution temperature. So the lowest critical solution temperature depends, or LCC depends on hydrophobicity. So if you look at this patchy system, has hydrophobic patches, has a very low LCSC, and this one over here that has no hydrophobic patches has an LCSC of around 60 degrees. So before the 60 degrees, before the LCSC, everything is in solution. The solution is clear. But after the LCSC, everything comes out of solution. In our case, we form the hacky sac after that point. So in this case, as I mentioned, the one that has no hydrophobic patches, it has a very high LCSC, while the one that has a very prevalent presence of hydrophobic patches, it has a very low LCSC. So the more hydrophobic a system is, the less energy it needs to come out of solution. So now that we have this process down, then we look into what are the SdQs, or guanosine quadruplexes. The quadruplexes are based on tetrads that stack on top of each other, tetrads of 4 guanosine derivatives that are held together by hydrogen bonding and stacked together by the help of a cation, in this case potassium. So we synthesize our guanosine quadruplexes, we synthesize our guanosine derivatives, we add some potassium salt, and then we increase the temperature to form the hacky sac. And the problem here is that we need a lot of salt. We need 2 molar concentration of salt. And that is not that great for biomedical applications. So we look into the Maxier series. The Maxier series gives you an observation of how an ion behaves in solution. If you look into your left of the series, you have strongly hydrated anions, which are cosmotrophs. These are anions that prefer to interact with water molecules. But in the case of the right, these are chaotrophs. These chaotrophs prefer to interact with the solute because they have weakly hydration. So they end up serving as bridges between the water molecules and the solutes. That would mean that you would need less salt of chaotrophs to make your system soluble in water. So as I mentioned, in the process of salting in and self-assembly, you will not need this 2 molar high concentration of salt, and more data of what ended up happening is in the poster at Twitter. So please don't be afraid to contact me on Twitter. My Twitter is trial_errors, and my email is maxier.acosta@upr.com. Feel free to contact me. I'm excited to meet you all. Bye.